Okay, let's finish out this chapter. The third part of this chapter 14, we're going to talk about pedigrees. So pedigree is a family tree that describes parents, children, grandchildren. It can go across generations. And when you do it, you're going to come out with something like this. So males are going to be squares and females round. So here you have a father and a mother, these two people married and then had these children. So these are these two people you're looking at, their parents' siblings, and their grandparents. And whatever trait they're looking at, so here they're looking at a widow's peak, here they're looking at if they can taste this chemical called PTC. If they have that trait or that ability, it's going to be colored in, and if they don't have it, it's going to be left white. Okay, so here she has a widow's peak, she does not. If you look at their parents, they both have a widow's peak, and if you looked at their grandparents, you'd see a mix. So you are just creating a pattern. So you and your siblings could also look and see if, whatever trait you want to look at. If you have widow's peak, if you can taste it. So here she can't taste the PTC, here she can, it's bitter, and so she's making that face. So you just can go back and see who has that trait. If among you and your siblings, if you have children, you can put your children on there. Your spouse, what your spouse's trait was like. What your parents' siblings, and with their parents. So you can trace patterns of inheritance, you can trace particular traits, and kind of describe them using a pedigree chart. So, if you're worried about having a certain disease, you could trace who in your family has the disease, and if it comes down, and you can figure out how likely you are to have that disease, how likely if you had children, how likely they would be to have a disease. So pedigrees can be used to make predictions about future offspring. It's not going to tell you your child will be this or won't be that. It can just kind of help you figure out the probability of a specific phenotype showing up in the next generation based on how you can see it showing up in your past generations. Okay, let's also talk about some recessively inherited disorders. So many genetic disorders that we talk about are inherited in a recessive manner, right? We're not going to have a lot of dominant diseases because that would mean anyone with that gene would be more likely to pass on the disease. So having it recessive means you're likely to have carriers. Genetic disorders can go from mild to life-threatening. So here we are looking at albinism. Albinism is a trait where you don't have pigment in your skin, your hair, her eyes. She is very she doesn't have pigment compared to her sister. So if you have two parents who are both heterozygous, they are both going to be carriers for the disease. They're not going to express the disease. Their children can either have two copies of the dominant allele, which is the normal typical phenotype. They could be carriers. So I've said that word a couple of times. A carrier is a person who has a normal phenotype because they have that dominant allele, but they are carrying the recessive trait. So they could pass on that disease to future generations even though they don't have that disease themselves. So you can see two different ways that in a heterozygous monohybrid test cross, you could have two different ways that you could create a carrier. And then 1 in 4 of their offspring is going to have albinism, this trait, because that is a recessive trait. Recessively inherited disorders only show up if this individual is homozygous for that allele. So you need to have two copies of that recessive allele to end up having that trait. If you have any dominant alleles, then you are not going to have that trait. Okay, so here's again, carriers are heterozygous individuals who carry the recessive allele but are phenotypically normal. So she could be a carrier or she could have two copies of the dominant phenotype. Most individuals with recessive disorders are born to carrier parents. And so these carrier mom and carrier dad have that 1 in 4 chance of having a child who is afflicted with whatever symptom or syndrome or disorder that you are looking at. If a recessive allele that causes a disease is rare, it's unlikely that two carriers will meet and mate. So for a lot of diseases, they're going to be, for a lot of genetic diseases, they're going to be rare if that phenotype is, if that if that allele is rare, because it's unlikely that you're going to have two people carrying that recessive trait that would meet and marry. But that likelihood could go up if we are talking about consanguineous matings. So a consanguineous mating is between two close relatives. So when you have two close relatives, it's more likely that that both of those parents of the child would have the same rare allele because they have the same genetic history. So that is why you sometimes see in cultures or populations that have had a long line of consanguineous matings, or inbreeding is kind of the more common term for this, that you can see some rare diseases sometimes accumulate in those cultures. And most societies and cultures have laws and taboos against marriage between close relatives, right? It just feels squicky, like, to think about having cousins, first cousins, or even second or third cousins, like getting together. It just naturally feels kind of weird to us because of that genetic drive to have partners that are different from us, so that our children are less likely to have diseases. Okay, cystic fibrosis. This is the most common lethal genetic disease in the United States. So we're not talking about diseases spread through like germs, we're talking about genetic diseases. Cystic fibrosis is the most common lethal disease in the United States. About 1 out of 2,500 people of European descent develops this. The cystic fibrosis allele results from a defective or an absent channel, so a chloride transport channel. Without this chloride transport channel, or if you have one that doesn't work, you're going to end up with a lot of chloride channels outside of the cell, unable to get in. And they're not able to get in, so the cells aren't quite functioning, the system isn't quite functioning correctly. As those chloride ions build up outside the cell, some of the symptoms you end up seeing are mucus buildup in internal organs and abnormal absorption of nutrients in the small intestines. Before I had children, I'm of European descent, so is my husband, so my OBGYN suggested that we get tested. I got tested for it to make sure I wasn't a carrier, and if I had been, they'd make sure that he wasn't, so that we would know, what are our chances of having a child with this disease. Because that mucus buildup, if it builds up in your lungs, you're not really able to breathe, and that is very fatal very quickly, right? We need to breathe. And you're not getting good nutrient absorption, so you're not able to function as healthy as you normally should be able to. Sickle cell disease is another genetic disorder. This affects 1 out of 400 African Americans. And what happens is you have a substitution of a single amino acid in a protein, this hemoglobin protein, which is used by red blood cells, and that affects the shape of your red blood cells. So in sickle cell disease, you have this shape of your red blood cells. If you're homozygous, all the hemoglobin you make is abnormal, so all of your cells are going to have this. And that is going to lead to you feeling weak, anemic, pain, fever, organ damage. If you're if you're heterozygous, you are going to have half of your red blood cells will be kind of the sickle shape, and half of your red blood cells will be this more normal round shape. So you are going to have some red blood cells that are still going to function normally to carry oxygen to your body and carbon dioxide away from your body. And so you're going to function pretty normally unless blood oxygen is very low. So if you're like hiking high in the mountains where your oxygen concentrations are lower, you may develop some diseases. What's interesting is when you have sickle cell, you're less likely to have malaria symptoms. So these red blood cells can't be infected by the germ that causes malaria. And so you're protected against malaria. So in certain situations, malaria or sickle cell could actually be advantageous to you because it protects you from malaria. In some situations, having this more traditional shaped red blood cell is more advantageous because you have better oxygen flow. So we see places where like high in mountain ranges, like in the Alps or somewhere, sickle cell is not genetically common. But places where malaria is indigenous, where malaria exists and you're likely to encounter malaria, you see sickle cell disease is more likely there's more likely to be a genetic history of sickle cell disease. Okay, so this is what I just said. Pause and read this slide if you want to. Okay, and then dominantly inherited disorders. Some human disorders are caused by dominant alleles. One example would be this particular type of dwarfism, achondroplasia. Achondroplasia is formed as a form of dwarfism caused by this rare dominant allele. Dominant alleles that carry a lethal disease are rare, and they're typically just going to be a mutation that happened in that individual. Because if they're lethal and you have a dominant allele, you're going to die before you have children, so they're not likely to pass on. So they are going to be rare. But you can see if you have two heterozygote parents for this, the different phenotypes you can end up with. Oh, sorry, this is if you have one dwarf phenotype and one normal phenotype. So a dwarf father with a normal height mother, their likelihood of having that phenotype show up in the next generation. And also the timing of onset of disease can affect its inheritance. So if you have a disease that is fatal before you have children, it doesn't get passed on. But if you have a disease that is fatal, but it doesn't become fatal until after you've had children, then that disease can be passed on. One example would be Huntington's disease. Huntington's disease is a disease that generally just breaks down your nervous system. It's a degenerative disease. So instead of generating your nervous system, you are breaking down, degenerating your nervous system. But this disease doesn't have many obvious phenotypic effects until most people are 35 to 40 years old. So generally by the time someone is 35 to 40, they've probably had a child by then or had as many children as they want to by then. And then they find out they have this disease. Then after this age of 35 to 40, they start having symptoms. And then at that point, the nervous system begins deteriorating, and it is fatal. But because it's not fatal until after they've had children, it can be passed on. Many diseases, so heart disease, cancer, alcoholism, mental illness, have a lot of genetic impacts and a lot of environmental components. And so because they have so many factors playing into them, we don't know exactly a lot about each of these diseases, although they're being studied. But no matter what our phenotype, your lifestyle has a tremendous effect. No matter what your genotype is, your lifestyle has a tremendous effect on your phenotype. So no matter what genes you may have to predispose you to heart disease, if you are eating a heart-healthy diet, you're going to be a lot healthier. If you're eating your whole grains and vegetables and you're staying away from a lot of sugar and salt. If you stay away from alcohol, you're not going to develop alcoholism if you just never drink it. If you are doing things to protect yourself from cancer, if you're wearing sunscreen, you're less likely to get skin cancer, no matter what your genotypic predisposition may be. So you can make those choices to minimize your effects, even if you do have genetic predispositions to things. Like, just take those diseases seriously and make the right healthy choices. Genetic counselors, this may be a career that is interesting to you. So genetic counselors are professionals who can provide information to prospective parents who are concerned about a family history for a specific disease. So if you've done your pedigree and you're worried about something, you can go to a genetic counselor. I talked about how I took a cystic fibrosis. I had my genes, to see what my genes were for cystic fibrosis, if I carried that gene. If it had come out saying I had that gene and my husband had that gene, we could have gone to see a genetic counselor to determine, what our options were. So here's just a case study. A couple both have a brother who died from the same recessively inherited disease. So these two unrelated people. Wife has a brother who died. Husband has a brother who died. They both have a brother who died of this recessively inherited disease. So a genetic counselor can help them determine what risk they have of having a child with a particular disease. And important to remember that each child is an independent event, so its genotype is unaffected by the genotypes of older siblings. So just because you have a sibling who has it doesn't mean you have that gene in your body. And just because you have one child with a trait doesn't mean that other children have that trait. Okay, so in this case study, if both members of the couple had a sibling with a recessively inherited illness, both of their parents had to be carriers. So each of them has a 2 out of 3 chance of being a carrier, right? So overall in that 1 out of 4, we already know they don't have the disease, so we can take that 1 out of 4 out. So you're left with 3 individuals, three chances. 1 in 3 chance of being homozygous for not having the illness, and 2 out of 3 chance of being a carrier for being heterozygous. So if they're both carriers, then there's a 1 in 4 chance that each child would have their recessive illness. So the overall probability of having a child with an illness is 2/3, which is the chance that mom is a carrier, 2/3 is the chance that dad is a carrier, and 1 out of 4, which is a chance if they're both carriers, how likely they would be to pass on 2 recessive genes. So they have a 1 out of 9 chance of having a child that has this illness. Okay, so you just say 2 times 2 times 1, 2 out of 3 times 3 times 4, 36, which comes out to be 1 out of 9. Okay. For a growing number of diseases, tests are available to help identify carriers and help define the odds more accurately, so you actually know better than this general vague sense. Tests can enable people to make more informed decisions about having children, but they also can raise other issues, so whether affected individuals fully understand their genetic test results and how the test results are used. There's a lot of ethical areas, some things that are just flat up not ethical, and some things that are kind of murky in their ethics that we still kind of as a society have to grapple with where we're going to fall in developing these tests and how they should be used as these tests are developed. Okay, let's talk about a couple of different types of testing you have. One is for a fetus to be tested. One is amniocentesis. In amniocentesis, so the baby is in amniotic fluid. The developing fetus is surrounded by amniotic fluid in utero, in the womb. And if you draw out some of that fluid, that is amniocentesis. You can take that out. Chances are that baby has cells from that baby that are in that amniotic fluid. So you take out the fluid along with whatever baby cells are in there. Already in the fluid. You're not taking it off of the baby. You just cells that are in the fluid, and you can test for different diseases. In chorionic villus sampling, and you can see this baby is much younger, much less developed, so this can happen a lot earlier. In chorionic villus sampling, you take a sample of the placenta. The placenta is developed by the baby, has the same genetic profile as the baby. So by taking the genetic profile of the fetus, or by taking the genetic profile of the placenta, you know what the genes of your fetus are. There's also ultrasound, which can kind of measure different body parts and measure fetal health in utero, but does not actually give you a genetic profile. It just can assess if they appear to have any phenotypic effects. Okay, and then after a baby is born, there are also newborn screening tests that are done. So babies, when they're born in a hospital, or even I had babies born at home that they had this heel prick done. They just prick their heel, take a sample of the blood, and test for it. All states in the United States have to do at least 5 tests for 5 different diseases. One common one is fetal ketonuria. Fetal ketonuria is a disease that is pretty rare, 1 out of 10,000 to 15,000. But it affects a baby's ability to break down a certain amino acid. And so if you know that baby has a disease, you just don't allow them to eat anything with that amino acid in it. You are very careful about what that individual is able to eat, and they're able to live normally. But if your baby had PKU and you didn't know it, and you allowed them to eat things that had that amino acid in it, they would very quickly develop some mental and physical handicaps as a result of that. So by testing for this at birth, we are able to just completely protect those babies who have that so that they have an appropriate diet. Okay, so that is the end of chapter 14. If you have any questions, as always, feel free to email me, message me on D2L. We can talk in office hours or talk, anytime that you need to talk. Any questions, and I will see you next time. Bye.